Good morning, everyone. I am Dr. Tenzing. I am head of department in forensic in STNM hospital. I have been in this field for almost like nine years now. 4 years in Bengal and 5 years in Sikkim. So we'll be starting on this topic, recent trends in DNA examination. What are the advances? What are the impediments or the drawbacks? How the report writing is given, and what are the how it's put up in court as evidence. So before we start with the recent trends, we'll first start with basic intro about what is the DNA, because if I go into the recent trends without knowing the basic part, you will not be able to understand much of how the DNA is analyzed. Usually, as a layman, most of them think that a sample is collected from the scene, or any sample is taken, you just put it in the DNA machine and the result comes out. But that's not the process how it goes. It's a very lengthy process and it's not fully automated also. It's so much, almost more than half the work is manual. It has to be done manually. So first we'll just start with the intro. So DNA evidences plays a major role, a key and essential role in today's modern criminal justice system. So whatever nowadays, usually most of it, maximum number of cases we see every time something is sent for DNA analysis, be it in case of paternity, be in case of civil cases like paternity, inheritance, or be it in criminal cases, everything is mostly sent for DNA. Now for our state, since we don't have the DNA testing which is done here, so usually we send it out of state to any other central forensic science lab or any other state forensic science lab. And there's a huge backlog there. That's one of the drawbacks which we'll talk about, because those states have their own number of cases. So when we send, we normally, the time period is almost like 6 months, 7 months, even more, depending on the each CFSL or state FSL where the sample is sent. So one of the most scientific tools in investigation and court proceedings, it helps identify and exonerate individuals and prevents conviction. Indian courts increasingly rely on DNA evidences and emphasis on reliability and chain of custody to ensure evidence is scientifically sound and legal and trustworthy. So reliability now, just because we send a sample to any DNA lab, so the entire process of sending those chain of custody, how the sample is collected, those all have a SOP. So if the standard operating procedure is not maintained in the first part, whatever the result comes out, usually it's not accepted or it's not taken. So we'll talk about the DNA, simple basics. What is the DNA? Right? So we see, whenever we say the word DNA, that image comes in our mind, right? So now the thing is, we all as different species, now main thing is, as a human species we have 46 chromosomes, right? 23 pairs of chromosomes. We get 23 from our mother and 23 from our father. So one differentiating factor is only the humans have 46 chromosomes, 23. Other species, like usually we see like other species, sometimes maybe animal blood or any other species blood may be mixed, right? If it's there, the number of chromosomes segregates which is human and which is any other blood, right? So humans have 46, that is 23 pairs, each from the 23 from the mother and 23 from the father, right? Out of 23, Twenty-two is called autosomes, and it's almost similar to everyone. Like you, me, any one of us. 23 is almost, we have almost similar kind of. Now that will differentiate 2 eyes, hands, all those factors. But out of this 22 also, few minute changes will be there that differentiates the person's ethnicity. Maybe a African, maybe dark skin because of these changes in the genetic factor, right? So Asians shorter, maybe that one factor. Some people have brown eyes, some people have blue eyes. So that may be some minute changes, but almost maximum we all share the same thing, right? The 23rd chromosome, which is the differentiating factor, is a male. That's called the sex chromosome. If you are a female, you have XX, and if it's a male, you have XY. So the 23rd chromosome is only to differentiate, or we look at that only for the sex difference. Okay. Now, when we take for a DNA testing, when we do a DNA testing, we do not do specifically for only this or only. We take the entire 46. Entire 46 we take it, right? So out of the entire 46 chromosomes, what is there is 99.9% of chromosomes is useless. But it's same for everyone. Only 0.1% of the chromosomes is unique, and that differentiates everything. Now we are just playing with that 0.1%. I think you're getting it, 0.1%. Only 0.1% is the differentiating factor. That's why whenever we get samples, you see, like whenever we get samples like old samples, degraded samples, mixed samples, it's very hard to determine the DNA of a particular individual because differentiating factor is only 0.1. That whole sample may be having lots of DNA chromosomes, but 99.9% of it is useless, which is similar to everyone. So 0.1% is the only thing we are looking at. Now, thing is, now 46 out of the 40. Now, since we have this, right? This is a chromosome, right? So there's few terms I think which will keep on coming up. So if it comes up, I'll just explain it to you, so it will be easy. So there's, you can see the word written locus, right? Locus means, in short, you can say location. So this is a DNA. This is from my father. This is from my mother, right? And locus means location. Now the DNA has, this DNA is, if you see in the image, there's a like joint at middle, like there's a bridge type. And at the end of each bridge, there is a letter. And only 4 letters, entire DNA you will find only 4 letters. Those 4 letters are A, T, G and C. No other letter will you find in a DNA. So you see that therefore humans have 46 chromosomes, 46 strands. Each chromosome is 1 strand. And in 1 strand means this is a 1 strand, this is 1 strand, and in 1 strand you have millions and millions of these letters. Now it's not possible to look at everything. That is not possible. We don't. It's not. Even the newer techniques cannot do that. So we are when we do DNA testing, we look at particular segments only or particular locations. It's preset. Beforehand, it is decided that we'll look at 15 locations or 20 locations, and whatever sample comes, whatever samples we get, it's pre-decided that we'll look at location number 1, 2, 3, 4, 15 locations we'll look at. So maybe this is the location we are looking at, 1, this is the location 2, this is the location 3, and it can be like, then the other chromosome, right? Other chromosome, that location, we have particular location for that, we are looking at that. So, and when we look at these locations, what we look at is, these particular locations, we look at these letters. So, this bridging thing is there, no? So, when it's one over here A, then it will come T, over here G and C, okay, A, T. So, it makes a pattern like A, T, G, C, A, T, C, G, all these words it comes, right? Till here, I think you are getting it clear, no? Till here. So, now, since we get these patterns, now we'll look at location, this location. Let's say for my mother, this is location number 1. Similar thing, my father, location number 1 is this, for my father. Now, over here, location number 2, this is for my mother's side, location number 2. Now, what will happen is, for each location, we see the number of repeats. Will come, I'll show you to the next slide. Now, loci means location, we are looking at location. Gene means small section of DNA with instruction, like A, T, G, C, all these are genes. Alleles means this is one allele, this is from my mother's side and this is from my father's side. So each chromosome has two alleles. One is from father, one is from mother. So similar to this one, you see the spiral structure. First one is the chromosome. Now the spiral structure has been unwinded and you get those letters. A is binding with T, C, G, T, A. So it's one horizontal, vertical line is one allele, the other one is another allele. Now I'm looking at the number of repeats or letters. Let's make it like first on this side, on the right hand side we see A, G, T, C, A, T, G, A. Other side we see T, E, A, G, T, A, C, T, right? So those letters are formed. Then when those letters are formed, there is something called STR or short tandem repeat. The name itself says, no, short repeats. So maybe the letter, since like it's like ATGC, suppose 4 base pairs ATGC, and again maybe after some time it's repeating itself, ATGC. So in that segment, that location, how many times ATGC has been repeated? Maybe 10 times. On my mother's location number 1, I have ATGC repeated 10 times. And on my father's location, maybe it may not be ATGC, it may be GATA. So number of repeats may be 9 times. So first, when the DNA result comes, chromosome location number 1, ATGC 9 repeats and GATA 4 repeats, right? So this locations for a DNA to match, when you have two samples, the minimum criteria as per the US has different criteria, we have a different criteria. We need at least 15 locations to match. 15 locations. So we are looking at 15 locations. This and this is location, same location, 1, 1. So this makes one location. So I am getting a result for one location. There are 9 repeats in my mother's side and 4 repeats in my father's side. So this same way, location number 2, location number 3, 4, 5, 6, we'll have, everything will come in numbers, how many repeats has happened. So when you cross match each 2 samples, the result has to be 15 out of 15. All 15 locations exact it has to match. And in our country, one more thing is added, that is something called, they have added something called 15 locations has to match along with a protein, along with a marker called amylogenin. So amylogenin is basically a marker for X, Y or XX chromosome. It's a sex chromosome marker. It will just tell you that person is a male or a female. That's it. So you get DNA match, then you get a match of the sex. So it has to be a complete match. So sometimes what happens is you may not get 15 matches, you may get 14 matches out of 15. So that result doesn't confirm a 100% match. It may be you can say it's a partial match. So that time you may have to not only depend on the DNA result, you have to look at other side also to see. Less than 4 matches, that is a complete no match. Less than 4, if its location is matching, only 4 locations are matching. 4 locations are matching with each other. Rest out of the 15 and 11 is not matching. So that's a different sample. Or it may be a degraded sample, or it may be a mixed sample. So these are the drawbacks we'll talk about later. So short tandem repeats. Nowadays, whenever these are different terminologies, like different, this STR segment, STR word will come up very much because whatever testing is being done at present also, we are using only short tandem repeats. We just look at this STR repeats, number of repeats in my father's side, mother's side. I see the number of repeats, I compare it with the other match. So let's see how many match. Out of 15, 15 should match. That's okay. Now variable number tandem repeats. This was used earlier. Now this obsolete. Okay, this is like they used to use like long segments, because that time the machines which we had, we used to do everything manually. Nowadays at least we have a DNA machine which does half the work is automated. Earlier everything was manual, so that time you needed to, since it's a manual thing, you needed long number of letters to at least identify her. So these are obsolete. And that time the technique which was used is gel electrophoresis. Then SNP, a single nucleotide polymorphism. Now this STR and SNP will come up every time. Maximum states CFSL, RFSL, wherever we use STR. Now SNP is used, it's a very latest technique, and there are only 2 labs, if I am not wrong, in our country which can do this type of testing. One is in NFSU in Gujarat and one it's in Chandigarh. So this is a total, the machine which we use is total high-tech machine, more expensive, more manpower, more training is required, and it's the softwares are expensive. So these kind SNP and all, where it's used is when there is a mass disaster. Like suppose let's consider this recent Air India crash was there in Gujarat, right? So multiple victims, 204 or something like that. 204, 210 people died. Now you have to identify through DNA. STR, it's impossible to identify. STR, if you have more than 3 people's blood mixed or 3 people, it's very difficult to identify or give a result. Because STR is usually for up to 2 or 3 you can differentiate. More than 4 samples if it mixed, STR cannot differentiate. Now SNP can differentiate large number of samples, very large number of samples. So that's why majority in mass disasters, all these we use this SNP, single nucleotide polymorphism. Single means in SNP we are not looking at number of repeats. In SNP we do not look at number of repeats. So I'll just let's assume this ATGC, ATCG. Let's assume this one. This is one person. So another person, maybe the other sample, I'm taking of another person, a relative or someone. I want to cross match whether, suppose let's take the same scenario. I found a body in that Air India crash, and I'm cross matching it with another relative to see whether it's, so maybe ATGC, ATCG. So it may not be the same, the entire alphabet. You look at only single letters. So there will be a chain of letters, and that time also the location of which letter to choose is prefixed. So I'll look at chromosome number 1, location number 2. Suppose I'm looking at that letter. So I'll look at that letter. So if there is a C here and a C here, that matched. Now I'll look at randomly. I'll look at chromosome number 3 and location number 2. Okay, so that matched. So those locations is not chosen by me, it's prefixed, it's chosen from beforehand for everyone. Like in short tandem repeats, 15 locus has to match. In SNP, 50 to 100 letters has to match. Then it's a match is similar. Okay. We see in law, in court related cases, the majority, everything we get in STR. We have to see, we get this STR form, we don't get this SNPs. This SNP, mainly they use it for, not much in investigation, they use it, like I said, just for this mass disasters, to segregate all these extremely decomposed samples, for those cases. Now, there is something called mitochondrial DNA also. Now, mitochondrial DNA is what genes I am getting from my mother. Only from my mother, not from my father. So this mitochondrial DNA also, the machine which is used is the same as SNP, like those 2 in Chandigarh and I said in Gujarat. It's called new generation sequencing machine, high-tech machine. So that machine does STR also, that machine does SNP also, that machine does mitochondrial DNA also, that machine does YSTR also. It does everything. Whereas the machine which we use, which all the states have, rest of us, is that machine or that method is called capillary electrophoresis method. We'll discuss on that later, because that's the most which is being used, capillary so that will use anything, all methods only if this STR, like normal STR it will use, and it will use this YSTR. Any other letters you see, mitochondrial DNA, SNP or whatever it is, if there is no STR in that, that machine cannot do that work. So mitochondrial DNA is mainly through this, in that newer generation sequencing. If you look at this, I can just trace my maternal lineage. I cannot go towards my father, you cannot trace it. So if the mother wants, if the mother is the, you are cross-matching someone's with the mother, then you have an option not only of STR, you have the option of mitochondrial DNA also. You can do a STR also, you can do a mitochondrial DNA also. Now, if the match person whom you are matching is with your father, so or you want to see this person is the father, so that time you can do only two methods, that is normal STR or you can do YSTR. YSTR is same like STR, but you are looking at the STR repeats only in the Y chromosome. You are not looking at the X chromosome. Okay. So mitochondrial DNA is found in mitochondria passed from mothers to all children. So maternal lineage you can see. It's useful in degraded samples, hair, shaft and bones. Mitochondrial DNA testing uses sequencing, new generation sequencing, not STR. So benefit of doing mitochondrial DNA is at least I don't have to look at all the segment repeats. I just have to look at mitochondrial DNA from the mother. I just that portion only. And the second benefit is nuclear DNA which is passed on to the sibling or to the son or daughter. You just have two nuclear DNA, one from the mother and one. But mitochondrial DNA are like thousands and thousands of, so lots of DNA is passed on to the, so you have more number of DNA to look at. Now YSTR, so short tandem repeats found only on male chromosomes and used for male specific identification. So that YSTR you can look only at the, you are looking at the YSTR with more of male, related to male towards the father's lineage. So they are STR markers on the Y chromosome. So if I am looking at YSTR and I want to see my father's lineage, I can go towards it. So YSTR is used by CE, that is capillary electrophoresis. That is the method which we use right now. And serology is like before DNA, what other tests used to be used, like blood grouping, all those come under serology. So pre-DNA biological fluid tests, blood, semen, saliva, like semen test you do, blood test, saliva, urine, vaginal fluid. Use chemical and immunological tests, not DNA, anything other than DNA. Now, something called DNA typing versus DNA profiling. So simple typing means making a DNA pattern. All this work which is being done, like I am making this work, right? I am separating the locus, this much repeats, all the work which is done is typing. Profiling means at the end when you get the result, you cross match. Does this match with this person's profile? So that's the thing. So DNA typing, examining specific DNA regions to create a unique DNA, and profiling is using that result which comes out to identify or match a person. Now samples. What are the samples which can be taken for DNA analysis? Blood, saliva, semen, urine, any of these samples can be taken. Hair, with or without root. If you have root, with the root is better. Then we have nails, teeth, bone, any muscle tissues, personal items, clothes, toothbrush, skeletal remains for disaster or old cases. So all these samples can be taken for DNA analysis. Now, usually so many people ask, like for skeletonized or getting a blood, saliva, semen, urine sample and all from a fresh body or a living person, it's easy. Sample collection is very easy. But in degraded samples, in skeletonized samples, what are the samples which should be picked up and whether that sample is good enough to get a result, those are the queries which comes up most of the time. So when it's a skeletonized samples, we usually choose long bones, like bones with marrows, maybe like my femur or humerus, long bones where I get a marrow, or I choose we choose usually the teeth, especially the molar teeth. Okay, since there's a pulp inside, so more chances of getting that. And if in degraded samples, other than if you don't bones, what are the samples is, if it's a degraded tissue and all, we try to pick up the entire muscle tissue. So these are the two things. Now result depends on how degraded or how old the sample is. So it varies from each case. Now differentiating human versus animal blood. So let's assume there's a crime scene, blood is splattered all over. So whether first assumption is whether the sample is a blood or not. Whether it's a blood or not, that is the differentiation factor. So there are few tests we do to determine whether it's a blood or not. So one is the Kastle-Meyer test and one is luminol test. These two presumptive blood tests will tell you this is a blood sample or not. But it will not, it will not determine whether the blood sample is a human blood sample or any other species blood sample. That cannot be determined by this test. So this will just segregate it's a blood or it's a paint or it's a any other thing. Then confirmatory test will decide species. So this will tell you it's a human blood or it's not a human blood. So these are the two tests which will tell you, that's precipitin or ELISA test will tell you it's a human blood or not. Species identification. Now once the species is identified, okay, it's a human blood. Now tell me this blood is of this person or not. So that time first you do grouping. If grouping matches, then we can go for DNA, because grouping alone cannot identify. Then we go for DNA analysis and we see the blood matches there or not. So that's the. Now other than test, we can just do a simple microscopy also to determine whether it's a human blood or not. Because human blood are usually, there's no nucleus and it's biconcave. Cells are usually biconcave and the rest of the mammals are nucleated cells, right? So second factor is, if you skip, you just do the first test and instead of confirmatory test of different species, precipitin and ELISA, I don't want to do, I just want to go direct DNA analysis also. I can determine whether it's a human, it's a species differentiation can be done, because as I said in the beginning, humans have 46 chromosomes and other species will have different. Maybe like monkeys, apes, monkeys, apes have 45, I think so. No, 45. So other species will have 20. So different, based on the chromosome number also, differentiation can be done. So workflow is, first we collect the stain, we check if it's blood or not, Kastle-Meyer's. Then we come to the second part, human. If it's identified as a human blood, we go for precipitin and ELISA test to identify whether it's human or not. And lastly, we do a DNA profiling to identify particular individual. Now, evolution of forensic science is, over a period of time, how has this DNA come in? What used to be done previously? Previously, we used to have only classic serology tests like blood grouping and all was there. Blood grouping, protein markers. So these were the tests to identify. We didn't have this DNA and all these stuffs. So DNA came into 1980s. After that, we started having. First, we used to do everything manually. Gel electrophoresis technique. Everything was manual. Then we came into STR and mitochondrial DNA. Why STR brought higher resolution? We came into this part, and now rapid and probabilistic methods, imaging globally. Now rapid methods of DNA also are there. Many countries, especially the West, use it, but in India we don't use this rapid DNA technology. We'll talk about this later, why we are not using it, though it's much more easier. You get a result within 90 minutes or 90 minutes you get the result. But why don't we use it? Now evolution, let's not talk about everything, I'll just touch the important ones. So 1900s we started with blood grouping, 1900s. Then we came over to let's come over to 1980s. So 1980s we started with this DNA testing, manual DNA testing. Manual DNA testing, we call it RFLP also, that's a manual thing. Right now we are doing this STR techniques, right? So 1992 we came up with this YSTR, and mitochondrial DNA was used in criminal cases. These testing were used. And then 2001 SNP and microbial forensics. 2001 we came up with this new generation sequencing. But just now also it has not come up to each SFSL or CFSL. It's just there in 2 places. So this is a brief horizontal timeline. First we started with serology only, like before also I said, just blood grouping and all that stuff. 1980s we came into DNA profiling, STRs, YSTR and all those stuff. And now the latest one is new generation sequencing from 2000s, but the gold standard still remains the middle one, that is STR profiling. That is everywhere, we use this everywhere. NGS has come, it came in 2000, but still we just use it for that mass disasters and highly degraded samples. That also usually in high profile cases and all, they send it to NGS. Otherwise everything is done in STR basis only. Okay. So importance of DNA in judicial decisions is it provides objective scientific evidence. It corroborates witness testimony or circumstantial evidence. It can identify unknown or decomposed bodies and supports both criminal and civil litigations like inheritance, paternity, maternity, that comes under civil and criminal. Now one thing is there is usually when many people come to give samples, right, they say your sample CFSL pathaunu parcha because it takes a long period of time, right? Not mostly for civil cases. When they come to take civil cases, usually people come to take samples, right? They come to the hospital, so you have to take samples. That they say criminal cases, all the sample has to go to CFSL, RFSL state, right? So civil cases, they say since it's a long waiting period, they say can't it go to a private lab or something like that? So there are private labs actually for this. There's a private lab in major cities, Hyderabad, Delhi, even in Siliguri they take samples. They don't have the lab there, they just collect the samples and they send it to, there's something called DNA FS, forensic lab or something, private labs, chain of private labs. Now the thing is, DNA testing, the labs, they should have proper accreditation, validation, they should be recognized, and there's a particular standard which has to be maintained. Now it depends. Some say that samples have been taken to those private labs and labs have given the reported and some courts have accepted. Okay, some say that. Some say that the labs gives false accreditation. The government has never given any accreditation to those labs or validation. You can do those tests. And whatever result, though the result they may be doing the proper process, but the result cannot be recognized because it's not a recognized lab. So that dilemma is still there, and which I am also not so aware of because I have not seen such private labs reports here. But the thing is still there. Now DNA profiling. So this may look like too much, but I'll just try to simplify it. So one is DNA profiling means now you are trying, so sample collection. The first part is sample collection, collect biological materials containing cells. So let's, all those samples which was there, urine, blood, all those sample collection part comes here. Okay. Now then comes DNA extraction. Now whenever any sample is taken, it's not that, like I said in the very beginning, it just, when someone says DNA testing, it just looks like, let's take a most common sample, the blood. So you take blood sample of 2 people and you put it in the machine, you close it, and the result comes out, match, not match, peak, see. So that is not the case. It's a very lengthy process and more than half of the thing has to be done, still it's done manually. So when the sample is collected, any sample, let's assume any sample, DNA extraction means, let's take blood. Now in that blood there's proteins, there's, other than DNA there are so many substances, proteins and unwanted substances. Now I want only the DNA, because if I put the entire sample for testing without extracting just the DNA, the results may come faulty or it may not give a result. So there are methods to extract the DNA. Now the latest method which we use as of now, DNA extraction, is it's called magnetic beads method. Okay, so by the magnetic beads, the DNA gets separated from the blood sample. Only the DNA part comes out. So that's a purified DNA sample. That is first part, that is the extraction part. Now quantification and quality check. Now once the extraction has been done, I get only the DNA from the blood. Rest of the samples, proteins or whatever is there, I leave it. That's not required. So now when I get only the DNA, now I have to quantify and quality check. So quantify and quality check depends on the sample, purity of the sample. So if I'm assuming I'm taking a sample blood from one person, so I'll get a very good quantity of DNA. After extraction, I'll get a very good quantity. Now same thing, suppose I'm taking a degraded sample or a very old sample or contaminated sample, whatever it is, not fresh sample. So I'll get a very less amount of DNA because it's already mixed, or more than half of the thing is already decomposed, degraded. So I'll get a very quantity, my initial quantity will be very less. So now, and then quality check, whether that sample which is there is enough to give me a result later. That is one check has to be done there. Now coming over to target selection. Now when the sample is taken, like I said, the target is already fixed. I have to look at 15 locations and my target is already fixed. I'll look at chromosome 46 chromosomes. So I'll look at chromosome 1 this location, chromosome 2 this location. The target is already fixed. So target selection already is there. And the target selection, how they do is they use something called primer. It's like a color coding. Let's like say I want to look at location number 1, red color primer. I'll already put a red primer. This is my, so I'll know that red means I put in location 1. 2, blue color, 3 colors. So color coding will define my locations. Then comes DNA amplifications. Now I need only those targeted locations. Rest of them I don't require. So rest of them, so it's like cutting only the targeted locations and taking out, and rest of the whatever is there you leave it. So now the targeted locations, since the sample, when the sample is given, you have the DNA machine requires a more amount of DNA to give a result. So what the DNA amplification means, as the name suggests, it will amplify. So you have this target location which I have taken out, it will amplify, it will make like 100 copies of this. So when I cross check later, when I do my testing, for location number 1 I have 100 samples. Let's assume like doing testing on a single piece of grain and a handful of grain. Right? So testing will be easy for a handful of grain. So I'm amplifying it, I'll make it more. So I get a better result. If it's a degraded sample, maybe that handful of grain, whatever, even if you amplify it, the core DNA is degraded or not good sample, then you will not get a proper result. Then DNA separation and visualization. Once it's amplified, then we'll visualize the particular DNA. Then DNA sequencing. Then we put in the machine, whichever machine we have here, capillary electrode machine, and it will sequence it. It will take out the STR segments for each, and then we get analysis, bioinformatic analysis of the result. We get the result, and then it will interpret it, and lastly reporting. Now the few drawbacks, people say why not everything should be done. Now till the sample collection, extraction, quantification, target selection and amplification, all these are done manually. Manually in the sense, there's a sample extraction, quantification, from target selection and amplification there's a separate machine for that. And DNA testing, sequencing, there's a separate machine for that. So it's not that we have only one machine for DNA. There are like 2-3 machines. And from transfer from one machine to another machine has to be done manually. So let's see the first picture. This first picture is of the earlier thing, which is now we don't use that. Both are capillary electrophoresis method. The second picture is the one which we still use in SFSL, CFSL. Maximum we use this one. So let's see the first picture. Extraction, restriction enzymes means you just whatever, say DNA sample, whatever pieces are there, that are the locations which I have selected. So I want those samples only, rest I don't want to, because I have lots of samples in the restriction enzyme, it's a long sample, I'll leave the rest of it. Now I'm just looking at the DNA sample which I have taken. Now electrophoresis is the method by which the samples are arranged. And at last, then transfer to membrane, incubation, these all were done manually, because that time there were no machines. At last they used to have a X-ray or a digital fingerprinting. They used to take an image in an X-ray form so that it doesn't get lost, to keep a copy of it. So at last they used to match it. I'll show you in the next slide how they matched. And those samples used to, we'll see in the image later, it comes in lines and colors, like the color of the primer which we have chosen before, it comes in. So when two samples are kept together, the color and the length of those matches. Now the technique which we are using is the STR method. So sample collection is done, DNA extraction, you can see it's all manual, DNA extraction is manual. Then quantification is done, machine, that's a separate machine for that, quantifies how much sample is there, it will tell you the purity of the sample, what probability, probability of the results coming 100% or 90%, 80%, will tell you that. Then we come to the multiplex PCR amplification, that's just another machine. So we have second machine that will amplify your chromosomes, whatever samples required will amplify. And then DNA analysis, this is the third machine. And then it will give you data result. So whenever the result is given, the machine gives the result, but it has to be cross-verified manually. Again, later it has to be cross-verified manually. Is there any, did the result come out properly? Is there any drawbacks or faults in that result? So that we have to cross-check at the end. So flowchart is, once evidence is collected, preservation and chain of custody. We all have a standard operating procedure for this, how the evidence is collected, how the entire preservation and chain of custody has to be maintained. Then comes the lab part, receipt and registration. Then we do this DNA extraction, amplification, quantification. Then we come to the profile generation, electrophoresis. Electrophoresis is the method which is being used. Okay, latest as of now we use this only. It's called capillary electrophoresis. Earlier it was gel, now it's capillary electrophoresis. Gel electrophoresis, they used to differentiate the DNA based on the length of the DNA. Now capillary electrophoresis, what benefit it has is segregation of DNA strands is done on length as well as charge, whether it's a positive or negative. So charge, so we have double advantage. Now the interpretation and statistical evaluation, STR, and then report writing, and then court presentation of DNA evidence, judicial evaluation and court decisions. Now types of DNA analysis and tests. So autosomal, so what, like I said before also, what in DNA testing, what we look at. So we'll look at this only, STR, which is the most important. Routine cases, high discrimination. Maximum we use this, STR, only STR. Why STR? To look at my father's lineage, of if it's a male. Okay, why STR? Male specific sexual assault. So like, suppose assault case, sexual assault case, you find a sample of blood or semen and there's a suspected accused. So now cross-matching that semen sample or the blood sample with that accused, I can do a STR, but why not narrow it down and bring to YSTR? Since he is a male, why not just do a YSTR? Suppose another case, let's assume you have to cross-match it with a female. So sample, we have to do a STR. Over here we just do STR because mitochondrial DNA and SNP I cannot do because I don't have the resource. The machine is not there. Now for that you have to send all the way to NFSU, Gujarat or Chandigarh, which I don't think so. Usually we don't send also. So we just do the STR and we'll separate it. But it takes a longer time and it's quite difficult than YSTR. YSTR is easy. I'll just look at the Y. Right? So mitochondrial DNA, degraded samples and maternal lineage. SNP is the most high-tech one. So 50 to 100 letters has to match at particular locations. So people say, no, this person is the 10th generation of this one, 7th generation. That can be determined only by SNP. Now, why not? People say, why not STR? STR does the same work. Now, thing is, my father and mother has passed on their genes to me, right? So 50% comes from my father, 50% comes from my mother, right? So my probability of matching with my father's DNA is only 50%, and with my mother is only 50%. Now same way, my matching with my grandfather, it goes further down, 25%. Now again another one, it goes on down to 12%. Same with me and my brother. Though we share the same parent, is not going to be a 100% match. I get my DNA 50-50 from mother, 50-50, but we are not the same. He may be taller, he may be a little bit bigger, he may be more fairer. So genetic variation is always there. So basically it's like 80 to 85% match. Now identical twins, exact twins, match is very high, 99%, almost 99% match. So they say that if you have to choose for uniqueness of a person, fingerprint is much better than DNA, because if it's an identical twin, match is 99% is a quite big match. So you cannot, sometimes the lab also 1% lab may also miss, right? Even if it's not an identical twin, he is my sibling. We have a 80-85% match, but still the match is quite high compared to others, right? But the fingerprint, whether it's an identical twin or no person, it cannot match with anyone. So that's why the fingerprint is still not obsolete. It's been there for very long time and still we use it. Now rapid DNA technique, quick on-site testing. The rapid DNA technique, I'll elaborate it later, but it's being used in the West. It's just how they do it is, they just take the buccal sample. They don't take, they find someone, they just put a cotton swab around the mouth and they put it in the machine, and within 90 minutes it gives the result, DNA analysis, it gives the result. But the drawbacks also we'll talk about later, why it's not being used in. Now criminal applications of DNA analysis, where the DNA analysis is mainly used. So sexual assault and rape cases, number 1. Homicide and violent crimes, 2. Linking suspect to a crime scene, blood or touch DNA. Okay. So that's why they say, at the crime scene, do not touch everywhere. Then identification of decomposed or charred bodies. Now one thing is, usually they say skeletonized body, DNA you can get, you take the bones. Decomposed body also, either you get the bones is last to decompose, so you get the bone sample, that is always there. Teeth sample is always there, because, and muscle sample maybe you find a little bit. But what about burnt bodies? So let's say in case of burnt bodies also, the entire thing is burnt, but we usually notice wherever a body is burnt, the last thing to be burnt is usually the teeth, especially the molars. It will always be somewhere or other you find some molars, third molar or second molar, you find those. So that one teeth is enough to give a DNA sample, number 1. Now, though it's difficult, very difficult to get those samples, but if you find a molar teeth or any long bones, you will not find. If you find just the molar teeth, I think that result should come out. But result will come out mostly in the form of likelihood. It will be written like chances are there, but the likelihood is this much, or the probability is this much. It will not come out like perfect match, something like that, because the sample itself is a very old degraded sample, right? So civil applications, paternity and maternity disputes. Paternity, I'll just do a YSTR. Maternity, if possible, a mitochondrial DNA. If that I cannot send, then I'll do a normal STR. Inheritance and succession claims also. Inheritance also you can, SNP is one of the best to check inheritance. Now paternal lineage, YSTR. Insurance claims and compensations to identify a particular person is a relative or not. So YSTR cross match or STR cross match can be done. Then disaster victim identification. So that if it's a mass disaster, then number of casualties, then it has to go for SNP. Otherwise, if it's just 2-3 people, then STR can be also done. Now special circumstances. Degraded DNA from old cases, difficult. It's difficult to find get a result. Mixed samples are multiple contributors. Mixed samples are multiple contributors. If you send samples to state labs, if there is a sample of mixed, like I said before, more than 3 people, result will be difficult. Up till 2-3, result may come out. But if the sample contains more than 3 people, then it's difficult. That can be differentiated only if you send to NGS, okay, newer generation sequencing, to Gujarat or Chandigarh. Exhumation for cold case investigations and missing person identifications. Now one drawback about NGS is, like the example which I gave in the beginning about that Air India crash, there were so many victims, everything was done in NGS, SNP, they did a SNP because STR is not possible. Now the probability thing is, they gave the results, but you will still find that many people or many relatives said later they found that the body they received or whatever they received is not there, is not theirs. There are many articles written that particular person later did this, that, but the body which they found is. So NGS also doesn't give 100% result. The mix may be there. It'll just give in a probability or likelihood ratio, but 100% result, that also doesn't signify. That's why we still say that the gold standard test for DNA is STR. We'll use STR, gold standard test. Now, the test I said in the beginning, the gel electrophoresis test, the result comes out in the picture pattern. This nowadays we get it in graph, peaks, but earlier we used to have it in slides. So DNA sequence mostly identical among humans, because out of 23-22 pages, 99.9% we share the same. Only 0.1% is the differentiating factor. So majority it may look quite similar, the color coding, length. So polymorph region, now we are looking at this STR segment, SNP segment. These are only segments which are unique and we need to look at. Even siblings can be distinguished because it's not a proper, you don't have a proper match. It's a 88% match, and with monozygotic twins, identical twins, it may be around 98, 99. Exception is identical twins. So let's look at the diagram. It's an old gel electrophoresis diagram. So sample collected of the suspect. So that is the result which we got. Now we have suspect 2, 1, 2, 3. Color coding is the same because we are using the same primers. Now you can obviously make out that the suspect 2 is matching with the sample which is collected. It's exact. Right? So based on the length and the color coding, the sample is matching. So let's assume these are two individuals, so green and purple. The middle part which they share is 99.9%, that is similar to both of them, and only the 0.1% is the differentiating factor. So shared DNA 99.9% same among humans. Unique DNA is the 0.1%, where all the STR fragments and SNPs we look at this only this 0.1%. Now shared DNA is common to all, 99.9%, common genes, common sequences. Unique DNA, individual, first one, STR. There may be variations in STR, it may be a rare mutation. So not only looking at the law point of view, these genetic information like SNP can also determine genetic diseases, like particular genetic disorders. Like this person has a high probability of getting this disease. So that is also one thing. SNP differentiation unique to A and individual's specific genetic markers forms the personal DNA profile. Same in B. So same method, sample collection, extraction, PCR setup, we use one machine, quantification we use another machine, and thirdly lastly the sequencing we used another machine. So basically 3 machines are used. 3 machines are required to do the testing. Same sample, preservation chain of custody, DNA extraction, quantification, amplification, electrophoresis, interpretation. So sample collection. So before we go in this, I'll just say about the machine which is used for STR. Why STR is gold standard, not only on the testing point of view, but if you look at the cost factor also, training factor also. So thing is, STR machines which we have a STR machine in Ranipur also, but thing is and we have been having this machine since a very long time. If I am not wrong, it's there since 9 months or 10 months. But the drawback is there is no trained personnel who can do the first part. It's not, if it was just putting the sample, anyone will, but that first part of DNA separation, extraction, quantification, so that part no one can do it because they have not been trained. So unless you do that part till the PCR amplification method, then only you can put in that lastly to give the result. So that part no one is trained. So that's why we are lacking it. Now,
why can't they be trained?
No, they are now. Thing is, when I talked last to them, I think I just talked a week back. They told me that recruits will be sent next year, this coming year, 2026. The recruits are being sent to CFSL, and they'll undergo training. But the date from when DNA analysis will be started in our state is still even they are not aware of. So that is so they said hopefully by 26 it should start, but the trainees have not gone till now. So they have not been sent for training. So hopefully they are sending it 2026, but they are not aware of which month, which date, they are not aware of. That's the one drawback. And training they will be sending to CFSL. That's what they told me. So one thing is training factor. It has to be a well-trained person. The second thing is cost factor. Now let's bring it to the cost factor also matters. STR machine which we have, there are 2 types of STR. We don't call it STR machine, we call it electrophoresis machine. Capillary electrophoresis machine. There are 2 types, 8 and 24. Why number 8 and 24 is, in 8 you can put 8 samples and in 24 you can put 24 samples. Majority which we use not only in our state, just now we are sending samples to Kamrup, Bhopal, Calcutta, sometimes to Hyderabad. I've seen samples being sent there. Maximum everyone is using 8 sample machines, 8 slots, so you can put 8 DNAs and get a result. So thing is, 8 sample machine, that cost of the machine, only that machine will cost around 1.3 crores. So that is the one factor. One is that one cost factor. That's just the initial cost factor. Now second thing is AMC or maintenance of that machine is around 10 lakhs per year. That is another factor. Now the third thing is all those softwares which is being used in that is being updated every time. So it's like a software which we buy normally also. Upgradation of software. So each time software is upgraded, the cost factor, you'll have to purchase a new software. Okay. So same thing for cyber forensics also. You cannot stick to just one software. Every time the software is being upgraded. So that is one. Now third thing is reagent or polymers which are being used. Just for that, whatever the samples, reagents they put inside, so that also has to be purchased on regular basis. That is also one drawback. And thing is, I have 8 slots. I put one sample. Now, why the people, there is a bit of delay is because people wait for all the 8 samples, then let the machine be full, then I'll run it. Number one is, it'll save time, it'll save my resources, it'll save my manpower. So where some, like let's assume our state is small, so maybe 8 samples, you will not get 8 samples, you get 1 sample, but the amount of time, so even if you do all 8 samples also, the running cost of the sample, once you run it, around 6 to 10,000 is the running cost. Whether I put 1 sample or whether I put 8 sample, my running cost is 6,000 rupees. So if I put 8 samples, 6,000 divided by 8, my price goes down, right? So that is also one drawback which is being faced. So that is also, so if I have 8 samples and I put 8 separate times, maybe if I do one today, tomorrow, so 6, my cost goes up. So that is also one factor. So these are the drawbacks of this. Now coming over to why we are just sticking to this electrophoresis machine. Why not bring a new generation sequencing machine? It can do STR, it can, whatever the capillary electrophoresis machine can do, or new generation sequencing can do all of that. On additional, it can do a SNP, it can do a mitochondrial DNA also. So why not? So first is cost factor. Number one, it's almost 4 times the cost of capillary electrophoresis. It starts from usually from 3 to 4 crores. That is one. AMC double, 20 lakhs. Time period, STR machine, normally STR machine will give, if I'm not wrong, result comes out in 6 hours. One sample, if you put the result comes out in 6 hours. But new generation sequencing machine will take around 2 days, 1 to 2 days. That is also one factor. Second thing is, new generation sequencing machine has a slot of 96 samples. Now bringing 96 sample machine to our state, already 8 slots cannot be filled. Running that 96 samples is more costly, number one. And softwares, training, it's almost double, triple of all these. And all these machines are imported. We have there's a company called Thermo Fisher. That is the one who makes all these, not an Indian company. So all is imported. So that is also one drawback of this. So basic, in simple, it's like capillary electrophoresis. Like let's assume I have a book. Capillary electrophoresis will read line by line. It will take a long time, but it will read line by line. New generation sequencing will read the entire book in one go. But the problem is, new generation sequencing faults may be more. You are reading entire in one go in a short time. In 2 days you are reading the entire sample. So more mistakes may be there. But why STR is reading line by line, but it will take you a long time. Very long time. So that is also one thing. We'll talk about this later also.
Why can't there be a machine with only one sample?
No, I think there is no company which has made that. It's either 8, minimum is 8.
All over the world?
Yes. There is no single slot machine, there is no single slot. Minimum is 8. Less than that you will not get. That's the drawback. One we have here is also 8. I'll show you the image which I got from them. I'll show you the image also. Now that is
why can't we have that buckle one?
That is buckle one is rapid DNA technique. That machine is a portable machine, but they are so. I'll tell you in detail because that machine also has various drawbacks. And second most important thing about all these is licensing and accreditations. All these like not only this DNA labs, all private labs also which we see normally for health, which is all over. They have this NABL, National Accredited Board licensing, and there's something called ISO, International Standard of Organization, all those license, right? So rapid DNA technology, no license, no accreditation. Newer generation sequencing, licensing is one, though it's there, but it's not scientific. They say it's like it's not being used much. So licensing and validation and all, that is one thing. But this capillary electrophoresis method which has been used, and licensing, accreditation, validation, they have everything. So that's the reason why it's going towards that also. That is also one reason. So sample, I'll just do this slide, then I think we'll have a break. I think it's being too much overloaded, no? So sample collection, all the samples which we have taken, right? So thing is, that time I showed you all fresh samples, like you take a living subject sample blood, but sometimes maybe a blood stain may be there on the floor. It's not a liquid sample. Blood stain may be there on my coat. So that time what happens is, when it's a sample like that kind of, or semen sample may be there. So when all these samples which cannot be extracted easily, either for blood stains, what we do is, we usually, if it's a floor, if it's there, we usually try to scrape it off, and you get those clotted dust type of thing. Then we mix a little bit of NS or saline, and then we make it into a liquid form. That gives the sample, number one. Now same thing for coat and all also. If you have to, either if it cannot be scraped, like in coat the blood sample cannot be scraped, so either they take the entire coat, or either they'll cut that portion of the sample which was there, and then later in the lab they'll remove that blood stain. So that is one method of doing that. Now stored other samples, hair samples and all that you can take easily. Okay. Then bone samples, usually of dead bone samples, we prefer to send long bones, not sometimes if the tooth is there, you send the tooth, but we prefer to send the entire long bones. But so many times the forensic labs, they refuse, outside they refuse, they say sample is too big. So what we do is, normally we take up till 10 centimeters like that, we cut the bone from both ends, because they are mainly concerned with the marrow. That's what they are concerned with. Otherwise you could have just sent ribs or any these kind of bones, but they want these hollow bones usually. So if this is, we usually send 10 centimeters like that sample, or we send the tooth. That are the two samples. And for mutilated or not mutilated, let's say decomposed samples, we try to, if we can separate a particular tissue, we try to send. Otherwise we try to send a lot of samples, not just one, because at least if this doesn't go, this will go. This doesn't go, this will go. So recently there is one case, if I remember, just this year only. It's just, I think that case, there was a landslide in Chaten, in an army area. So there was a husband and a wife with a child, and all three got washed away in that landslide. The entire hill came down. Husband, wife, both were army officers, and the child was gone. Now the thing is, what happened here is, the father and mother came all the way, they are from Delhi, so the father and mother came all from Delhi, and they gave their blood to cross match, because the lady who died, she was their daughter, right? So to cross match. Now child, when they later on the army, all this was done by the army. The entire hill came down, they are just digging. They found the body of the child, and with the clothes and all that, and a little bit, they identified that this is the girl. The male person also they identified. They found it, and due to the clothes and all that, wallet and whatever, they identified. Now the problem is, they couldn't find the woman, lady. So every 15 days, I think till now, we just keep on getting samples. Sometimes they send, whatever the army people, they just dig and they just find, like, they cannot say, no, that this is a human bone or whatever. Sometimes they bring like small bones, it's like a bird, dead body of a bird bones like that. But they just send the samples. It still comes in STNM, it still comes right now, every 15 days and all. I think they dug that entire hill for almost 1 month like that. So that sample keeps on coming, and the father mother problem is they want the sample to be sent to Hyderabad. They were saying to the police that please send the sample to Hyderabad because the Hyderabad CFSL in charge is a relative of this, and the sampling testing will be done faster. So, but the issue is how much ever sample we send, it's never getting matched. So, in fact, in that when the landslide thing, we found samples of human samples, long hairs that cannot be of any animal or any human bones. Also, we found the sample. That's a human bone. We can make it out by the length and all that. But the samples is not matching. That means that bone or hair must be of someone else, but there is no report of any other person missing other than the 3 of them. So samples are keeping on going, but there is no result or output. I think till now samples keep on coming. It's a Lachen. It comes from Lachen. So sample collection. Now preservation and chain of custody. Stored samples in clean, dry, labeled containers, avoid contamination, document every transfer of sample, chain of custody, critical for court acceptance. Later on, when we come to the landmark cases of DNA, landmark cases of DNA, there have been several instances where the sample has matched. 100% match of the samples has been there, but since the protocol, a chain of system or chain of custody has not been followed, the case has not, there is no result to the case. But the person has been, suppose the person had to be convicted, he is free. So it's failed, even though it's a match, it's of no use. So we'll do this in the next, after the tea break. Start. So what are the various samples is done, then preservation of samples. That is second part. Now after the preservation of the samples, sample has to be sealed and all those stuff comes. Then chain of custody. Now from where the sample goes. So this is just a brief chart which I made. It's suppose let's take the sample. Crime scene is there. Police department collects the sample. Then the sample has to go to the forensic science department. Now forensic science department sampling. Either nowadays all the samples are picked up by the SFSL people. They call them on the site. They take the samples. And now the sample testing has to be sent, then the report comes back. The report now, what happens here is the report comes, of the sample comes back, then to the IO only of the case, then it comes to the court. That DNA sampling is, sample report comes that way. So now DNA extraction techniques. Now what are the DNA extraction techniques? Now when you get any sample, once it reaches the lab, how they extract DNA from that. You have to remove all the impurities, and also the method which is used as of now is magnetic beads method. I told in the beginning, there are various methods. Organic method is there which uses chemicals. Then Chilex method is there which resin, they use resin. Silica method is there, DNA sticks to a filter. Then there is magnetic beads method. DNA sticks to the beads with a magnet. And then salting out method is there, that is salt removed proteins. An automated machine does the step. This is automated. Nowadays everything is manual. DNA extraction techniques, everything is manual. Automated DNA, all these, like let's take a sample of a blood or any substance and then put it direct and all the result comes out in one go. That is fully automated. So there is no machine which is, as of now there is no machine which can do that, fully automated. The machine which we use here, the capillary electrophoresis machine, is semi-automated. Half work is done manually, half work is done by the machine. The NGS newer generation sequencing machine which we have two in our countries, that is also half is manual, half is automated. So at least you have to pick up and do that, put it in different machines. That is. Now fully automated machines is in process. They are trying out. It's called cartridge based. But just now what is happening is those are all in prototype. It has not been come out. It's in prototype and research work. And it has not been used outside also, in different countries also. They are not used fully in legal or any other things. It's just a research academic thing which is going on. So 2 out of these techniques we use only 2, that is magnetic and silica. Magnetic is the best one which is being used right now, and before magnetic we used to use silica when we were in that gel electrophoresis time. That time magnetic beads were not there, that time that banding used to come out, we used to do manually on X-ray plates and all that. That time we used to use silica columns. So these are the 2 how the sample mainly remove the DNA from the any sample. First part, extraction part. Now comparison table extraction, one is manual and one is automated, right? So manual is slower obviously, automated is faster. Manual hands-on, automated less use of manpower. Manual variable, you can make some mistakes also, but automated is very consistent, right? Contamination risk is higher in manual, automated is lower. Sample capacity, few samples, automated can do more samples. Cost, manual is extraction is cheaper, automated is expensive. Now you may be thinking that the only favorable thing which is going towards manual is the cheap setup. Rest everything is going towards automated, so why not fully automated kar dino? Why not do it fully automated and less, see, less, it's very, consistency is very consistent, low, faster, contamination risk slower, many samples going in one go, why not go towards fully manual? But the issue is there is no device or no equipment which is made which can do that till now, till date. And the one which is doing that right now, cartridge based, is in prototype, it's just in pilot phase. If I am not mistaken, once they did this, or they have this in West, cartridge based, once they tried it in Hyderabad for a pilot sort of thing, pilot project or something, then later it stopped because there were some scientific it's not fully validated, or like it's not been tested fully. So once the cartridge, in the near future, this is the, later in the future, maybe 10 years, 15 years down the line, this is going to come out eventually. Fully automated, it will go fully automated, but till now, we are not in that phase. Not only us, anywhere, any country, we are not in that phase. Now, extraction is done. Now, before we move on to, once the extraction is done, before we move on to amplification, to create more DNAs, why do we need to do the quantification? So, quantification will process, this process will tell you how pure your sample was or how much sample you had. Amplification will be done no matter what. Whether you have a degraded sample, whether you have a fresh sample, whether you have a very good sample, bad sample, amplify it will do, because small copies, you need multiple copies to make a, to give a result. But quantification will tell you how, what was your base sample, what was your initial, before the amplification part, what sample you had and how pure that sample was. That is also one factor which is required. Because just imagine, you have a fresh sample, I make 100 copies, and you have a degraded sample, I made 100 copies. Now the result of the degraded sample may vary, it may give a faulty result. So at least by that time you should know that the initial sample which you amplify itself was a degraded or a bad sample. The machine will not say that this is a degraded sample, I will not amplify it. It will amplify whatever you give. So, and quantification determines DNA concentration and purity. Now how when your sample is given, they'll say like how pure the sample was, how they determine how pure the sample was is fresh sample, you will get fresh, any sample you will get more number of DNAs. So I need to amplify it. Suppose I want 100 DNAs, 100 copies of DNAs. In a fresh sample I may get 10. So I'll just multiply it 10 times, I'll amplify it 10 times and I get 100 copies, right? Now, my target is 100 copies. Now I have a degraded sample. Maybe there's no 10 copies in that, there's just one. Because it's a degraded sample, very less sample. So to reach that 100 copies for that machine to accept it, I need to multiply it 100 times. So when you see that amplification number is more, that means the sample is more degraded. You getting my point? Pure samples, less number of amplification, 10 times sample, you'll give 100. Into 10, you'll give 100. If the sample is bad, you need to amplify it more. So the more amplification it does, because the machine requires 100 samples, no matter what. This is just an assumption. The machine requires 100 samples. No matter what is the base, I want 100. Now if my base is bad, I'll multiply it 100 times, be it I'll multiply it more than that also, 1000 times, but I want 100 samples. So the more I'm multiplying, my sample quality is that bad, that's why I'm multiplying it. If my sample quality is very good, if I have 50, I'll just multiply it 2 times and get my sample. So that's how the quantification or purity of the sample is tested. And that machine which does it is that process is called qPCR. It's also called real-time PCR, or PCR in short, if you extend it, is called polymerized chain reaction. It's a chain reaction which amplifies. It's called true PCR. So it ensures sufficient DNA for reliable amplification. It will tell you how pure your sample. Later on, what will happen is when the result comes out, you will have a good idea that since my quantification of DNA sample was pure, my result is better than the others. So it detects degraded or low number copy of DNA also. More amplification, the sample is that bad. These are the methods by which this quantification is done, or purity of the DNA is tested, and qPCR is the one which we used. Rest 3 are obsolete. We used to use it earlier, all this nano drop method, fluorometric method, gel electrophoresis, we don't use it now. So we use this qPCR, and it tests how purity is based on the amplification, as I said. So this is the machine. Now the drawback of this machine also is same like the capillary electrophoresis machine. You can see the number of slots. So they are like 96 or I think 120 chambers. Now whether I use one or whether I use 2 chambers, the other chamber has to be left vacant. So if I'm testing a DNA, I have a DNA sample, I want to amplify it. I'll not put it in all 96 chambers. I'll put it in one chamber and it will amplify it. So rest of that, whatever that blank thing is there, it's like a wastage. No? So if I have more samples, that's why people, what they do is, when you give the DNA outside also, why the waiting period is more? Number one, they have their own number of cases. Number two is, they want the entire thing to be filled. They want to run it in one dose with the full thing. If you run single, it's waste of time, waste of resource, waste of everything. So that is also one factor. So polymerized chain reaction multiplies the DNA. STR markers amplified using kits, this is amplified, and multiple loci analyzed simultaneously and critical for creating a reliable DNA profile. So you can see in that diagram, what that machine does is, the single DNA is made up into 4 copies of that DNA. Then after that is the electrophoresis machine, which will, the last final part, which will give the result. So capillary electrophoresis separates DNA fragments, produce an electropherogram showing adhesion. What it means is, earlier I showed in the image there were bands, right? So suspect 2, 3 matched with this. Nowadays that method is gone. That was manual method. Nowadays everything is computerized, digitalized. It comes in graph form. So let's assume that I'm looking at chromosome number 1, and the location which I have selected is location A for male and female, and I have given the markers which I put for location, that is red color. So I may get 2 peaks, red color peaks, 2 peaks. So I know that this is chromosome 1 which I have tested. So this may show 9 number of repeats. The more the repeat, the higher the peak. So the peak this peak is for one person, and another person, the peak has to match. Then you can say this is a match. And all peaks, 15 loci means 15 places and then male and female separate things, then almost 30 peaks will come out. So based on the graph, you cross match the two samples. You have to cross match. It's similar to that. That only thing is it will come in, that thing came in slides in colors. This will come out in graph. And the reason why I say more the contaminated the sample or more mixed the sample is difficult is because just imagine 5 samples are there and all are coming up in graph, red, yellow, green. Now what will happen is the graph will overlap each other. So it becomes so complex. The more the number of DNA is there of different people, the graph will become more complex. Maybe up to 2 or 3 people you can differentiate. After that the graph will become so zigzag with so many overlaps that you will not be able to determine who is what. So then the profile compared to reference or suspect samples. So you will compare that. Now the thing is, in capital area forensics machine, like that madam said, why not only one sample? Okay, that is also one thing. Now thing is, usually when any testing is done, to take out there's a blood sample or whatever sample, DNA is bound to come out if it's a human being. Now why we do DNA is either cross match with the suspect or with the victim. This DNA is this person's or not. So that person with whom you are matching also, that sample also has to be put. So 2 samples, and they put 2 control samples. So what happens is, in that 8 sample machine which is there, you can test for 6 people. But that 6 people, what happens is 6 people, 3 is the testing, 3 is like maybe the crime scene and all that, 3 people, but 3 is matching. You're getting my point? Like you are testing for 3 people. So when you test DNA for 3 people, you have to cross match with them. We have to link them with someone else, right? Either you put 3 people, yes, you get a blood sample, 3 blood samples in the crime scene, and 1 person, then you find one suspect, you say it may be his. So that time what you can do is put 3 samples of that crime scene blood sample and put that person's sample. So 5, and you have to put 2 control samples also to test whether the machine is working properly or not. So 6 samples and 2 is left blank. Now if I want, there are 3 separate cases, 1, 2, 3 separate cases. So this person I want to cross match with this person I want to cross match with this person I want to cross match with this. 6 samples. I'll put all the 6 samples. This person has to be cross matched with this with this. 6 samples and 2 control samples. So 3 cases is getting sorted out at the same time. But 2 control samples is always there to check whether the machine, whatever process was done before of amplification, quantification, that is proper or not. And those control samples check that, and the interpretation result which is coming out is proper or not. And those control samples, what graph it gives, it's already, because that has been done so many times, they already know that this graph is supposed to be coming out this way. So if there is any change in that control sample, that means something is faulty with the other samples. But still, if it's single case, like madam said, single case, 1 person and a cross match, and you put the 2 control samples, still 4 slots are going wasted. That is it. Now interpretation and statistics. Match versus non-match. 15 out of 15 loci with amylogenin, that is the sex chromosome marker, is coming out match. That means a perfect match. Less than 3 out of the 15, 3 matching, 12 is not matching, then that's a faulty sample. That sample is a no match. So anything above 4 or 5 up till 14 matching number, it partial match, and probability increases the more the match is there. Like 5 match probability may be less, 14 match probability may be like 90%, 95% match. So the result comes out in phase, it comes out like probability or likelihood, something like that. So match versus non-match is determined. Then likelihood ratio or random match probability. What could be the probable ratio that two samples match? Then consider mixtures. Now match, just because it doesn't match 15 out of 15 and 14 or 13, whatever matches, that does not mean that the two samples is not a perfect match or something. Sometimes either the samples are of two different persons, either there is a cross contamination, it's of the same person but there is a cross contamination in that, or the sample may be defective, or sample may be degraded or mixed. So that scenario is always there. It's not that just because it didn't match, it can not be of two different. Mixture can, contamination is always, that thing is always there. So expert judgment supported by software. Now once the result comes out, the result which graph is given is all automated. It's given by the STR mix or true arrays. These are the softwares which are used, which will give the result or graphs. Now why I say manual is, eventually when the control sample gives the graph and this gives the graph, you have to cross check. Though the machine itself will do also, but you cannot fully depend on that also, because it may go further. So for verification, once manually cross check also is done. That is also one thing. Now we come over to the recent methods of DNA testing. All the process is done. So capillary electrophoresis, we are using this one. Everywhere we use this capillary electrophoresis, and all those process initially all those process up till that amplification part is the same for capillary electrophoresis, is the same for new generation sequencing also. You have to do that manually. Different machines are there. Only the last part, machine which gives the result, capillary electrophoresis will give in STR, and this can give in anything. Any new generation sequencing can give in anything. Then we have new generation sequencing, then probabilistic genotyping, then mini STR microfluidic lab on chip. This is the one which I was saying it's a prototype which is fully automated but is not being used as of now. It's in research process. When this comes up, it's like direct sample, put the sample, direct everything automated, result comes up. No need to do any manual work. Automation and LIMS. LIMS is a software, automated, and rapid DNA technology that will come down. It's sort of a portable thing, and result comes out instant. 90 minutes, 2 hours, result comes out. Now capillary electrophoresis, I have already said. Now we'll go to new generation sequencing. That is the machine which is used. There are various companies. MI Sequence makes it, Thermo Fisher makes it, but all are outside international brands, and very, very expensive. The cost of the machine itself starts from 3 crores. Now it goes 3, 4, 6, 12, depending on more complex the machine. Number one, the cost of the machine is nothing compared to the running cost. The running cost will beat eventually the running cost will beat the cost of the machine also. That is also one. All softwares, reagents, very, very expensive. One time running 30 to 40,000 if I am not wrong. One time running of this. If you run the sample once. Capillary electrophoresis, one run 6,000 to 10,000. This running machine will cost 25,000 to 30,000. One time if you run it. Whether you put 96 samples or one sample, it does not depend. Your running cost will come out to 30,000. More the samples, you can break it down, lesser, cheaper, but very deep. Then AMC, then software, that is a separate cost. So very, very deep. And regarding this new generation sequencing, one thing is there in our country is there is not much validation or like licensing regarding this. So that's also one drawback, why this is not being used so much, apart from the cost factor. And one second thing is training of this is separate from that electrophoresis machine. This needs highly skilled. It's not like for here capillary electrophoresis machine, like Madam said, will be sending someone, CFSL person will get trained, come. But this training is totally different. It's like they do a particular course sort of thing. So what is done is reads thousands of DNA pieces at once. It's like reading a whole book in one go. Works on burnt old degraded samples. It can detect that also, because it's not only doing STR, it's doing SNP also, short nuclear polymorphism. It's looking at random sites for a single letter change, and 50 to 100, if it matches, it becomes a match. That is also one thing. Can solve complex mixtures. Gives STR, SNP, mitochondrial DNA all in one test. This machine can give all in one test. It's useful in mass disasters and unidentified bodies. It's more powerful than capillary electrophoresis machine which is being used, and is much more expensive, 4 times of everything, whatever this cost, 4 times into 4, running cost, machine cost, anything. Even time, this takes more. This takes around 24 hours, 2 days, 48 hours I think, to give a result. That capillary electrophoresis machine will give you result within 6 hours, the entire process. But this will give you in detail more samples also. It's used in special cases, not routine work. Routine work we do capillary electrophoresis. It's available in only few labs. That is NFSU, National Forensic Science Lab in Gujarat, and CFSL Chandigarh. So that is the machine, and result comes out somewhat this way. So I'll read so example of next generation sequencing result viewer. So result comes out, the main page of result viewer is composed of 3 domains. The 3 boxes are there. The first box will show you the basic test information. Second box will explain sequencing methods or other related information. What you did a mitochondrial DNA, you did a STR, you did a SNP, you did a YSTR, you will tell that. And the last box shows the mutation data with reporting result, whether it's a match, non-match. Now over here they are saying last box shows mutation data, means this was not done for cross-matching. This was done mutation data, means this can, like I said, to detect any genetic disorders or change in the genes. So any mutation in that also, changes in that also it detects. So that's why it's written mutation data. Now probabilistic genotyping. You can see those capillary electrophoresis graph will look something like that. The less the sample, the better the clarity of the graph. The more the sample, the more haphazard the graph will be. So probabilistic genotyping means computer assisted DNA interpretation. This makes it hard to separate who contributed which DNA. Number 1, some peaks overlap because 2 people have similar alleles. The graph shows DNA peaks from more than 1%. How it connects to probabilistic genotyping is, when peaks overlap and the mixture is complicated, humans cannot interpret it reliably. More zigzag, it's difficult to test. The probabilistic genotyping uses software. Now probabilistic genotyping uses software, and it will tell you percentage of this person, like a graph is zigzag, you can, multiple is there. Now you cannot say 1%, 2%, whose mixture. Now probabilistic genotyping software will at least give you a probable, like this peak is of suspect A, there is a 90 or 80% chance of likelihood of this being the sample matching. So probable will tell you that, it will not tell you an exact match. Less sample you will get an exact match. Analyze thousands of possible combinations, decides which DNA profile are most likely, and gives a statistical probability. It will give you a statistics, answer will come in statistics. Now mini STR and microfluidic labs on chip. Mini STR, very small DNA fragments are there, is required. It works very well with degraded or low DNA, amplified degraded DNA, very low DNA also. This is the future. This is the future of DNA. Microfluidic chips, tiny channels acts as mini capillary electrophoresis. Just now we have capillary electrophoresis machine this big. Now this is the entire thing in one small chip. For faster DNA separation, shorter STRs reduce samples and reagent requirements. So reagents and all less requirements. Reagent will not be required basically. Everything will be done by the chip. Together, mini STR runs easily in small channels, quick, reliable profiles from poor samples. It's useful in disaster victim identification and trace samples. It's mostly in, just now it's in research and prototype stage. Developed and tested in labs and academic studies. It's not widely used in routine forensic labs anywhere yet. Now automation and LIMS. Automations, whatever the manual which we are doing, picking up from one test, putting in another PCR machine, quantification machine. So automation means robotics and machines will do that, repeated DNA lab work. And LIMS is the software that tracks the samples, tests and results for accuracy and efficiency. It's a software which will test how the process is going on. So robotic extraction and quantity, these all are future, in the future this is going to come up. So robotic extraction and quantification, just now it's not robotic, it's manual. Laboratory information management system and entire digital chain of custody minimizes human error and contamination. So what will happen is, sample will be collected, barcode, each sample will be given a coding, automation systems, DNA extraction, PCR setup, amplification, analysis, all will be done in one go, one machine. Either that chip will do that everything, and then the data will be generated, and the software, LIMS software, tracks the samples and generates the result, and we can use that sample. This is later when that comes in, cartridge comes in place. This is it will go easy way. Just now we are doing it the manual long way. Now, like ma'am said, rapid DNA technology. This is the rapid DNA technology is being used in the West. Only buccal samples work. Buccal fresh samples work. Any degraded samples, drawbacks are degraded samples or mixed samples, result is not going to come. Fresh samples only. Now result comes in 90 minutes to 2 hours. Buccal sample only. Useful at police stations or border security, matlab instant, like it's like that breath analyzer types. Instead of sending your blood sample to the lab, just do the breath analyzer. It's not a replacement for confirmatory forensic lab. It's not a replacement for those capillary electrophoresis method. Requires strict quality control. Extraction cost per sample. One time running cost of this one sample is 12,000 rupees. One sample if you test. Same thing if you do it in capillary electrophoresis and all, sample will break down to 1500 or something like that. One sample. So cost is also one factor. Second thing is why is this not being used over here? So this second thing is there is no such for this, there is no license as of now. There is no license given for this. You need specific license and all that. One is cost factor, one is the license factor, and one thing is it's not being tested. It's just being used in the US, I think, right now. No other country is using it as of now. And even if you do this, you have to eventually go for the confirmatory FSL testing. You have to send the sample to the lab for the confirmatory testing at the end. So it's a long process, and even if you do this testing right now also, it's not accepted. Whatever the result comes is not accepted. That is also one drawback.
Has there been any incident where the result from this has not matched with the result with confirmatory?
This thing, what has happened is, since this is being used only in the West, we don't have any idea regarding how good or the efficacy of this machine is. That is also one of the factors why even after doing this, a confirmatory FSL testing is being set. And as of now, for this, no set laws has been put that this machine should be accepted, or this, since there is no licensing, this can be done and all that stuff. There is no such things. So it's not being used. So now let's just come rapid DNA and what rapid DNA technology and traditional methods like the ABI 3500C capillary electrophoresis is what we are using. That we are using rapid DNA profilers. So you can see rapid DNA is fully automated, no need to do anything. 2 hours you get the result. Electrophoresis 5 to 10 hours, and half of it is manual. So sample collection, extraction, amplification, all these steps in rapid is traditional is manual, and rapid is automated. In electrophoresis you can give any kind of sample. Rapid you can use only buccal sample, fresh sample, any degraded sample, no result is going to come out. So rapid DNA equals to cartridge for microfluidic systems. That to everything, basically the rapid DNA is that micro thing only, but little portable type thing. So it's being used automatically. In 2 hours it gives the result, and traditional capillary electrophoresis is a slower and manual prep, and it requires 5 to 6 hours for the result. Now we will come to the National DNA Database Initiative. Okay. So there was a Indian DNA Bill Regulation 2019. One bill was introduced. So as per this bill, what they had said is, let's keep a bank, DNA bank, one national DNA bank, and in each state let's keep a DNA bank, and in that bank we can keep DNAs of suspects or any person, offenders, repeated offenders, missing people, so that later on when we have any incident, when we have like a match, cross match to be done, you don't need the main thing, right? You can just put it in the system. The system will just, whatever sample I get, it will just collect from there. Whatever match is there, it will show this person, this person. Right now we don't have that system, so you have to find two people. Right? This is matching with this, you need two samples. So what they said, let's make a bank, DNA bank, and let's store everything in that. So that is being for, in US is already there. They have this bank. So what they did is, in this regulation bill, they are trying to follow the US pattern only. So US, they have this something called CODIS, Combined DNA Initiative System. It's a DNA bank, data bank, where DNA is, many people's DNA are there. So whenever you find sample, or especially suspects, offenders, missing people, these are the main people or people in the prison, these are the main people whose DNA is stored. So whenever you find any sample, you just cross match with anyone from there. If it cross matches, easier. So, but this bill was withdrawn in 2023. It didn't work out. So, where the clash happened was, they said this bill is against right to privacy. Article 21, if I am not wrong, right to privacy. You cannot just hold, even if it's a prisoner or whatever it is, you cannot hold someone's DNA against their will. No one is going to willingly give their DNA in the bank. So it's just, it may get misused or these sort of things. So this bill was scrapped off. 2023, it went away. It didn't work out. So that's why we don't have any DNA national bank or state bank. We don't have any bank for DNAs. Now CODIS is Combined Data Index System, US system for storing DNA profiles to aid criminal investigation. They have this. India plans a similar system like CODIS under the DNA bill-like framework. It had planned. CODIS is fully used in the DNA. National DNA Database Initiative existed on paper in 2019, but practically implementation is limited, especially at state level. It didn't work out, it was withdrawn. So what was the use of this? If this bill had come out, if banks had come out, what would have happened? It would have helped in identifying repeated offenders. It would have supported missing peoples and cold cases, and privacy and ethical consideration. This is where the main, why this bill got rejected and it didn't work out. So comparison, purpose of CODIS, criminal ID and forensic use, same in India. Database, they have a data, local, national, state, they have banks, state, DNA data bank, national. India had planned the same thing. Offenders, suspect, missing and unknown people, same thing. Matching, all was automated. So you just need to process everything as per this electrophoresis method, and you just need to, you have a, with whom you need to cross match, the bank will do that. So privacy was restricted access and same thing, restricted legal. Now we'll come to scientific challenges. So what are the challenges faced on DNA testing? So scientific challenges are broken or degraded DNA. Any broken or degraded DNA, not good samples, will give you a partial profile. It will not give you a good match or good result, number one. Low quality DNA gives you a weak signal. Mixed samples, that is also one drawback. Hard to separate contributors. Mixed samples, multiple, more the mixed samples, more the graph haphazard, more difficult to get the sample. Complex markers needs advanced analysis. Contamination, any contamination during the sample collection will give a false result. Now, inhibitors, PCR may fail. Now, till that part, contamination part, it's like sample itself was problematic. Now, sample is good, but the PCR machine which is amplifying, or the qPCR machine which is quantifying PCR, the machine also sometimes it may show a default. So that is also one factor which has to be considered. And if the amplification or quantification, whatever the machine, if it was defective, the control samples which we had kept at the end in that electrophoresis machine, that will go wrong, and you'll come to know that the first process itself was not proper. So the result will not be accepted. So alleys drop in, extra false alley appear, contamination, lab error, alley drop out. Alley means the two DNA sites, mother and the father, the location points. No, so this is one alley, this is one alley. So if there is contamination of another blood or any other thing, so AG, AT, a separate alphabet may come in between. So that will also disrupt that. Touch DNA. DNA from surface contact is unpredictable and may be mixed. So any DNA other than the blood, which is touch and all those stuff, those may be high chance of contamination in that. Now what are the technical roadblocks or drawbacks which we face? Technical, that was said to be technical. Now degraded DNA, heat, time, break DNA partial profile. So proper result will not come. So even if the sample was collected properly, you leave the sample just like that in heat or cold, sample is not preserved properly, the sample will degrade. Contamination risk, other people's DNA mixes with evidence, limited reference data for groups, laboratory procedure errors, then mixtures, multiple persons DNA overlap, hard to interpret. This also, alley drop in, drop out, same thing. Touch DNA, very little and unpredictable. Cost and time, advanced machine analysis are expensive. That is the last cost, running cost, we have to bear all that. That is also one of the drawbacks which is faced. Now, common operational problems, backlog and delays. Too many number of cases, less number of labs. It's not that, actually thing is, more number of labs can be made, but the licensing is one issue, number one. And second issue is, you cannot have a private lab doing criminal, for any criminal cases, the lab, it cannot be sent to a private lab. So that is also one thing. So that is one factor why there is backlog or too many delays. Number of cases, old cases, that result hasn't come out, so it takes time. Equipment failure and calibration issues. Second drawback, suppose there is a breakdown in that capillary electrophoresis machine, or if any error comes up, now you have to call that expert, since that machine is not from here, the person who is working, who is doing that DNA testing cannot make that machine. Now that machine will be, unless that expert comes from outside and fixes it, how many days, 6, 7 days, 10 days, 15 days, that all the work comes to a stop. So that is also one. Variable lack accreditations, different SOPs across different jurisdictions. So everyone has different SOPs. Sample contamination, that is also one. Degraded or low quality samples. Resource constraints, staff training and infrastructure. So this is the point where we are stuck. For our state, we are stuck in this point. Machine is there, but staff training has not occurred. So no one is able to use that. Improper storage and transport of samples. So legal challenges. Now let's assume all the testing has been done properly, result has come out, everything was proper. Chain of custody breaks, whatever the result comes out, no use. Sample becomes legally unreliable. Proprietary black box software, transparency and cross-examination challenges. Improper documentation, evidence challenge in court. Court expectations and statistical probability, judges law misunderstand. Like the problem is, I said, because sometimes result may not come out in just white and black, it may come out in grey, probability. So that time, who is the one who is going to decide what are the chances of this going to be yes or no? Because technically, the person who has done the test will give you in a ratio. Now, apart from that DNA, you may have to look in other sites, other places to corroborate and then come to a conclusion. That is also one factor, steps. So many times people say this match does not happen. Kati percent chance? No, kati percent. And the figure, sometimes the machine does not give a result like probability is 80%, probability is 90%. Machine may give probability may be between 80 to 90, something like a range sort of thing. So there it can be 80 also, it can be 90 also. Now which category does that fall into? So that is also one factor. So violation of privacy laws or consent issues, that is also one factor. You cannot, like especially in civil cases, there has been many instances like paternity, maternity, person is not willing to give the sample, and you cannot, without the consent, you cannot take the sample. That, though that's not for the criminal case, but civil cases that has happened. Just recently I was watching this Bihar elections also, so there was a news article which was news, one person was saying that during the reign of the Rabri Devi, when she was chief minister, her brother was involved in a murder and a rape case of a one guy and one girl. And later when, so this is a criminal case, and later when, as per the judicial, when the case was there, they said you have to give the DNA sample to for the case. He never agreed. Though it's a criminal case, it was said that you have to give your DNA sample for a cross match. It's not a, so he never gave the sample, and the case never moved forward also. The case just ended there, something like that. It was, I just saw it in one article. So that is one I didn't understand. You can, as per law, do you have the, can you just say in a criminal case that I am not willing to give my sample? That is also one thing. I didn't understand that part, because what I read is criminal case, consent is not required, you have to take, and civil cases you need consent. So privacy, violation of privacy laws or consent issues, privacy or ethical issues in investigative genealogy and SNP use, wrongful collection procedures, evidence excluded, expert cross-examination challenges, credibility challenges, lab errors, analyst and bias. So these were the drawbacks which is being faced. Now landmark cases on DNA evidence, I've tried to compile a few. So whichever I think I found out, I just put a few things. So this case was there, Santosh Kumar Singh versus State Priyadarshini Mato case, 2010. So these are the cases actually, Santosh Kumar Singh. Then we have this Nirbhaya case, 2017 DNA. Then Manoj versus State of Madhya Pradesh, 2022. Supreme Court guidelines on DNA chain of custody, ISO, that ISO 17025, that is the accreditation or required by the labs. So it has to be certified, international standard of organization, they are giving this number. So whoever has this ISO has the license to do that test. So then there is this Teku Datta versus Gautam Kundu DNA in paternity. It was a civil case, court sets. And then Swami Premananda case, confirmed paternity. This was one case. Then Nithari case, DNA identified victims plus linked accused. Then Tandoor case. This, out of all these, I think I personally, before I made this slide, I had heard about Nirbhaya case, I had heard about Nithari case and Tandoor case. Rest of them I hadn't heard about. I just, when I made this PPT, I just checked that and I heard about it. So identity proven by DNA of charred remains and conviction. This was the last case. So Santosh Kumar Singh versus State Priyadarshini Mato case, 2010. So what was the case was, Priyadarshini Mato was a law student and was stalked and murdered in 1996 by Santosh Kumar Singh, son of an IPS officer. She was found dead in her Delhi home with signs of sexual assault and strangulation. Now biological samples, semen, hair, etc. were collected from the scene. DNA profiling was done DNA and STR profile match the accused Santosh Kumar Singh. So capillary electrophoresis, that technique was done. It matched him also, the suspect. So match probability was extremely high, ruling out any other person. The Supreme Court held DNA as scientific, reliable and decisive. The DNA match became the key evidence leading to conviction. The Supreme Court set aside the Delhi High Court's death sentence, which he was given before, and set it to life imprisonment. And later this person got, in 2018 he was released after 22 years due to a court order. So this was the case. That white shirt, I think, was the person. So so I got this article. So this was the, this article, our brother or someone was requesting the Union Home Minister to not release him actually, something like that. Priyadashini Mato family meets Delhi Minister, urges him not to allow her killer premature release. So this is a 2018 article. So Nirbhaya case, this is one of the most famous things. So 2012, a 23-year-old physiotherapist intern known as Nirbhaya was gang raped by 6 people and brutally assaulted. Place was South Delhi, inside a private moving bus. Later she died from injuries. Biological traces, blood, semen, sweat, hair fibers were collected from the bus, victim and the accused. STR DNA profiling matched across victims, accused and the bus surfaces. Linked each accused directly to the victim and the crime scene. Court called DNA clinching and highly reliable, a pillar for conviction. And out of the 6, 4 convicts were hanged on 20th March 2020. One was a juvenile who served 3 years and then later he was released. And one person, he committed suicide. So, so this was the case where the 4 persons who hanged were, you can see in the image. So DNA played a major role in this also. Now Manoj versus State of Madhya Pradesh, 2022, Supreme Court DNA release. The case involved rape and murder of a minor girl in Madhya Pradesh. The accused were Manoj and 2 accused who were convicted for killing the girl. Murder case with serious lapse in investigation and evidence handling. How DNA helped us? DNA from the crime scene matched the accused and helped confirming the involvement. It matched. DNA matched. Now, why Supreme Court issued guidelines? It ensured to ensure proper DNA collection, sealing and documentation, to maintain chain of custody, to mandate use of ISO accredited labs. Only these labs, the results should be accepted. That's what the guidelines said. Purpose: to guarantee that future DNA evidence is accurate, trustworthy and admissible. They were still subjects to death sentence as initially imposed by trial court and confirmed by High Court. The Supreme Court did not comply the death sentence and allow hanging immediately. It instead directed a fresh hearing on the sentence alone stage, emphasizing the need to record and evaluate mitigating circumstances such as psychological or psychiatric assessment, jail conduct or background before finalizing whether the death penalty should be imposed or not. And the court felt the death sentence was given without collecting enough information about the accused's life and the mental condition. So the sentencing process was incomplete and unfair. That's why the Supreme Court stopped the hanging and ordered a fresh proper inquiry. So this was the article which I found for this. And there were many cases which were citing this. Later there were many cases which were citing this case also. So Teku Datta, Banarsi Das versus Teku Datta and Gautam Kundu versus State of West Bengal. These were civil cases, DNA and paternity cases. Mothers claimed that these were the biological fathers, but both the fathers refused to give DNA samples. Now verdict: DNA testing can be ordered only if absolutely necessary. Court must balance child's rights to know versus father's privacy. Test, if ordered, must follow strict safeguards, consent, all this lab, accredited labs, documentation. Outcome: DNA was not forcibly done. Paternity was not proved. Case set guidelines for safe DNA use. court held that DNA testing can be ordered only when indispensable, balancing privacy and justice. Supreme Court issued safeguards before compelling DNA tests. So these two people never gave their DNA, so it was not proved. Now Swami Premananda case DNA conviction. So he was the person. Now he is no more right now. He died when he was in prison due to some liver failure. He died then. But till his last breath, he always said that he was innocent. That was what was written in the article. So Swami Premananda, head of an ashram in Tamil Nadu, 1987-88, was accused of 13 rapes, 1 molestation and 1 murder. Murder of he was a male. Person who was murdered was a male. A female disciple became pregnant. Later DNA evidences, prosecution DNA test claimed that he was the father. Defense expert disputed this. Now what the thing was done was, when the prosecution claimed that DNA testing was done, prosecution claimed DNA matched. So prosecution claimed he was the father. Now what was done is the defense, I don't know, he got the sample and this sample was tested somewhere in UK. It was in UK, and then the result came that the 2 samples doesn't match. So he is not the father, something like that. But the 2 DNA reports contradicting each other. But the court accepted the prosecution's DNA as a part of evidence. Maybe since that testing which was done by the defense was in another country and not validated a licensed lab which follows our state's rules, so that was maybe the reason. So conviction and sentence, trial court verdict, he was convicted of rape and murder, sentenced to life imprisonment, double life imprisonment in some judgments in 1997. Appealed the Supreme Court and High Court upheld the conviction and sentence in 1990s, 2000s. Significance shows the role of DNA in criminal case, but also the importance of lab standards due to conflicting expert opinions. So this was the case. So Nithari case was quite famous because it was like serial murder case sort of thing. So the 2 persons who were accused were the last 2 persons, and the white shirt one was his servant, and all the bodies which were found in the drain behind their house. So they are free right now, they didn't get convicted. So Nithari case, when and where? 2006-2007, Nithari village in Noida. What happened was multiple children and women were reported missing. Human remains were later found in and around the house of the accused, especially in drains and all those. The accused Mohinder Singh Pandher, that guy, and his servant Surinder Kohli, domestic help, were charged with serial murders. DNA profiling was used to identify victims from skeletal remains and decomposed bodies. They could link the body parts to specific. They could link the body parts to the victims from their relatives, father, mother. They could link, but from the bodies which they got, they could not link it with the accused. That was one thing. From 2023 to 25, the Allahabad High Court and finally Supreme Court re-examined the entire case. They found DNA evidence only identified victims and not who killed them. Significance: DNA was crucial in identifying victims. These 2 people were acquitted by the Allahabad High Court in 2023 and Supreme Court upheld their acquittal in 2025. Investigation couldn't legally prove their guilt, so now both are free. So DNA could not link them. Now Tandoor murder case. So this person was the one involved. He was set free right now. He's free. Maybe he served 2 decades, almost 20 years, 22 years, something like that in the prison, and it was. So incident was in 1995, Delhi. Most of the cases all are in Delhi, India only. Sushil Sharma, a youth Congress leader. So he was a politician who shot his wife, Naina Sani, and tried to burn her body in a tandoor. DNA role, forensic expert recovered muscle tissue from the charred remains. DNA matched Naina's, the DNA sample matched with of parents, confirming her identity. Technical challenge, burnt remains, degraded DNA, success relied on bone or muscle fragments and PCR based methods. NGS was not available, so capillary electrophoresis obviously. And conviction, this person Sushil Sharma was convicted for murder, death sentence. Later commuted to life imprisonment in December 2018 by the Delhi High Court, ordered his immediate release after he had spent over 2 decades in jail. Significance, one of India's first case where DNA identified a charred body showing forensic science power even in extreme conditions. So this was the one of the first case where DNA evidence could be taken out from the charred or burnt body. Now DNA, now we are coming towards the end of the session. So DNA reports essentials. Case details. So what it should contain, the case details, the items, what was received, it should be in sealed condition and the chain of custody should be maintained. Methods and kits used, validated. All these extraction, quantification, STR analysis, it should be well validated, licensed. Methods, DNA profile, STR, peaks, male, female, mixture or partial. These are the different methods by which you can test it. Interpretation, either it's a match or a no match or likelihood or a probability ratio will come out. Statistical analysis, chance of random match 1 in million. Limitations and disclaimers, conclusion, match, non-match, inconclusive, expert sign and accreditation. So this all should be there in the DNA report. Now court acceptability is, court accepts DNA as reliable scientific evidence, reliable and validated method. Lab must use validated STR methods and be accredited, must have proper collection, sealing and chain of custody, follow all legal standards so the DNA report is valid in court. Report must be transparent, clear, profile, comparison and statistics. Clear explanation of statistical probabilities. Experts should explain results in a simple language. The court treats DNA as highly persuasive but not 100% absolute. It is not 100% absolute. So double check, it has to be double checked with other evidences. Compliance with ISO 17025. This is the license. Okay, the lab does its work properly and follows international quality rules, so the DNA test is accurate. So all this has to be there, then only the result is accepted. Best practice for labs and experts is strict SOPs and documentation for maintaining chain of custody. Accredited labs well licensed with quality audits, continuous training and technical updates, and collaboration with police, medical examiners and courts. Now, enhancing court understanding. So use very simple language and visual aids in that whatever the result is there. Explain the statistical probabilities clearly. So when you say like 80 to 90% chance, you try to explain it very properly what probability is there. Emphasize limitations. What are the drawbacks, or what are the if there is anything like mixture or degraded samples. I know you mentioned in the report. Avoid absolute highlight match probability. You absolute highlight match. You should, even if the report is fully it's matched, it says like 15 to 15 loci matched. It's a perfect match. It's just, but majority you cannot, even if it's a perfect match, you cannot write like 100% match and this sample is of this person, so is all those results you cannot write in that. So you just write it's a match. So judicial guidance and common questions is sample collection and storage. Sterile tools should be in that, all that tools which are used should be sterile and labeled container and proper preservation should be done. Handling of samples, the chain of custody should be maintained at every transfer. So this is the majority, sometimes chain of custody breaks, so the down part, whatever result comes out, no use. Contamination risk, minimized by SOP, followed all the standard operating procedures, protective gears, separate work areas, noted in report if suspected. Reliability of STR, highly reliable, more than 99.999% for 15 loci plus validated protocols used. So, like 15 loci is matching, still they are not writing 100%. They could have easily written, why to write more than 99.999, just write 100% match. But they don't write that. Statistical. We use 15 loci plus emelogenins. In US they use 20 to 25 loci. Their standard testing, they don't use emelogenin, they use 20 to 25 loci. They see 20 loci, more than that. So for them the match has to be more. Statistical match probability, random match probability or likelihood ratio calculated using allele frequency database software aids complex mixtures. Laboratory accreditations, all labs need to have this ISO 17025 accredited labs. Secondary transfers or DNA, secondary transfer rare but possible, addressed in interpretation and report limitations. So any secondary transfer DNA is any other samples getting mixed. So if there is, you have to mention in the report. Degraded or insufficient DNA, that also you mentioned. Techniques like mini STR, mitochondrial sequencing used, limitations required if unreliable. So degraded and in degraded samples, new generation sequencing is much better than capillary electrophoresis method. Obviously, because that's a high-tech machine, but limitations are always there. It's not possible to send every time. So mixed DNA samples. In mixed, it will come in statistical point of view, phenotype, probabilistic genotyping likely to assign likelihood of contribution. So it will give you a percentage, 80, 85, match, 90, 95, something like that. Reading the reports. Reports are written in plain language, explaining the methodology, what test was done. STR was done, why STR was done, SNP was done. Report will show that. Statistical probabilities and limitations. What are the limitations? What drawbacks were there? The judges are advised that the DNA evidence is powerful but must be considered alongside with other case evidences. So it has to be linked with other things. Now status of our state. This is the machine, exact machine. I got the picture from them only. So this is the electrophoresis, capillary electrophoresis machine. But before you reach this stage, there is two other machines. One is the quantification and one is the amplifier. Quantification machine is their amplifier. So that time when I said the cost of this machine comes around 1.3 CR and the running cost AMC is around 10 lakhs, and each time you put the sample 6,000, 10,000 rupees it goes, right? So that is the cost of this sample. Now what about the cost before this sample? The third amplification machine, qPCR machine cost around 4 lakhs. Running cost is different. Now before that there is another, not the amplifier, the quantifier. That small thing, that also comes around in lakhs. So cumulative, it's a very high budget. Apart, if you just see in financial point of view, cumulative itself is very high, and whatever the cost comes cumulative, maybe I said 1.3, let's add 4 more to it, around 1.7. Let's add 2 to 3 more than that for the quantifier. Or let's take it as 2 CR. The 2 CR is nothing compared to the running cost. Just imagine 10 lakhs you are paying AMC each year, and then each software is getting upgraded. AMC will give you will stick to the previous software. Now, like computer also be Windows 1, Windows 2, 3, 4, 5, 6, 7, Windows 11 we reached. So that software gets upgraded, the more upgradation it goes, the cost will go more. So it's said that, it was written that the cost of the machine and all these is nothing compared to the running cost. That running cost will beat that 2 CR down the line. So it's a very difficult thing for this one. So this is the machine which we have. Sikkim State Forensic Lab already has the 35 ABI 3500 genetic analyzer, 8 capillary. It's an 8 slot. 6 you can, 2 out of 8, 2 is always reserved for controls, and you have 6 slots. Now whether you want to put 6 samples 3-3 cross matching or that depends. It comes in two categories, 8 samples and 24 samples. You can have a 24 sample machine also, which we don't have it. And most of those where we sent also, Kamrup and all, they all use 8 capillary only.
What do you think as a head of forensic department? Should Sikkim have this, or is it better to send it to the other states?
Obviously, if we have it here, the report will come faster.
Keeping
easier.
in consideration all the financial condition also, what do you think?
I've seen, even the other states are using the same 8 capillary method. Now the cost, I think all the cost borne by the SFSL comes under the Home Ministry. So all the financial whatever is being spent is being sent by the MHA. The state doesn't have to bear the cost, number one. So whichever state it is, the Home Ministry Central is bearing the cost. So our state doesn't face any financial drawbacks with this. Now the thing is, if it's here, results are faster, number one. Though if we send outside, backlog is always there, always dependent. Now over here also, sometimes I'm seeing I don't know how they determine where to send the sample. Sometimes because sample, when they come for sample collection, the form has to be filled and it has to be signed by us. Because most of the time sample is collected by us, so we sign the DNA form. Sometimes I sign Kamrup form, sometimes I'm signing Bhopal form, sometimes I'm signing Hyderabad form, and it's not my choice that I decide. I never get to decide where the sample is sending. So once I ask them, sometimes you are sending Kamrup, sometimes you are sending Guwahati, sometimes you are sending Calcutta. So what they told me is, they asked Kamrup, then Kamrup said, we have a very high backlog, we cannot do your test. Otherwise, even if you send the sample, it's going to take a very long time. So then they asked Calcutta. Calcutta said, okay, we can give a little bit faster, you send. So that's how they decide. But that's what they told me. I don't know much about this, but that's what the IO tells. So we are sending different. So I feel if we have here, it's much benefit for us also. And I don't think so there's any loss for the state, because all the financial burden is being borne by the central government. So even if Bengal is getting 2 DNA testing centers, why not give Bengal one? Why give Sikkim one? So at least we don't need to depend on other states, because we are always requesting phase. We have to request. So DNA analysis has not started yet in the state. Eventually 2026 they are planning to send the recruits for training, then fixed date is not there, then tentative plan is to start by 2026 DNA mission. So new scientific recruits will be sent to CFSL next year for training in DNA extraction, STR typing and capillary. All that front part manual part, they will be trained for that. After training and validation, DNA testing is to be expected to be started by 2026. Exact start date is not fixed. That's what I was told. Until then, all current samples are being sent to CFSL or SFSL labs such as Kamrup, Bhopal, Chandigarh, Hyderabad, depends. These labs already receive heavy case loads from their own states, which causes delay in reporting for Sikkim cases. Obviously, when the case goes from here, they will try to sort out the case first of their state, right? So then they will do yours. They will not put you in priority. That is also one. So I think this is the last slide. So road ahead, expand DNA database with privacy safeguards, rapid DNA and automation for backlog reduction in the near future, capacity building and infrastructure upgrades, encourage early evidence collection. And the final takeaway is DNA evidence is powerful but not infallible. Value depends on whatever the result comes, the value depends on proper collection, analysis, transparent reporting and informed judicial evaluation. And coordination among police, medical labs and courts is critical. So I think this is the last slide. So now we can just have a discussion, like anything you need to ask, anything you have doubts, I'll be happy to clear it.
I have one.
Yes, yes.
How do you determine, with the DNA, is it possible to determine that the person is a product of incest?
No, that I think is very difficult with DNA. No.
Because half it comes, so 50% is from mother.
Yes.
So if the child is incest, because outcome of the incest relationship, that cannot be determined.
Very difficult. It's possible, but difficulty is very difficult. Very difficult. Anything else? Because what I saw is, whenever any test on DNA is done, everyone says though this NGS method has come in 2000 itself, this new generation sequencing has been there, and it can test so many things apart from STR. It can test your mitochondrial DNA, it can test genetic mutations, it can test so many things. But why we are just kept towards this STR only? Cost factor is there, that is understood, but cost factor may be fourfold. Like I said, STR, if it comes around 2 CR, maybe this is 8 CR, 10 CR, be 20 CR also. But the product which you are getting out of that 20 CR is far, far more than this. So, and whatever these two labs which is there, NFSU and Chandigarh and Delhi, whenever I see what that NGS is doing is only very, very high profile cases or disaster, take just to testing for this. It's not, apart from this I haven't seen anyone, till now I have never heard about anyone sending to, when I was in Bengal also I have never heard anyone sending to NGS, for NGS. Because sometimes what happens is, as it's said, right, degraded samples, STR is difficult. So like I have a degraded sample, STR fails. So what now? Shouldn't the sample be sent for NGS? At least, even if there's 10% chance of the result coming out, why not send it? But it never gets sent. That I always had in my mind, why is it not being sent? Till now I don't have the answer to that. But I always see in the news, mass disaster, NGS test is being done. Some high profile case, STR, not NGS is being done. So that I always had I had a doubt myself, which I wanted to clear. Till just now I have not cleared it. I have not got the result, answer to this. Why, which test which fails as capillary electrophoresis, why NGS is not being sent to NGS? Because eventually NGS is the future. Then once NGS comes over, then rapid DNA can be, rapid DNA or cartridge-based DNA analysis. We'll come to that later. That's a long way, long way. Because that cartridge-based is just in prototype, then it will come in the US, then after that, US it comes, by the time it comes, India it will take more than 20 years, I think. So that is one thing also. So any
What is the proper procedure in order to collect the samples from the accused? Do you keep the witnesses or?
So that depends on what sample are you collecting, number one.
Blood,
So thing is, what happens, I'll tell you what happens here and what happens in Bengal, I'll tell you there is no standardized form for that, number one. Even if you see any form, postmortem forms, postmortem reports, sexual examination reports, physical assault, injury writing reports, drunkenness reports, you will see each state has their own. Sikkim doesn't match with Bengal, Bengal doesn't match with Delhi. So nowadays, what the Home Ministry has done is, now they are trying to standardize all the reports into a single, so that even if one report goes from one to another state, it should be understandable and it should be same for that. So that Medlar software has come out, medical legal examination postmortem reporting software. So they have made that CCTNS for police, Medlar, I think Medlar, then e-prisons is there, then I think e-courts or something like that. So all fall under ICGS scheme, everything falls under that. So In that Medlar software, now that is being, as of now, Sikkim has started with Medlar. Postmortem reporting is being done and any other reporting is done through Medlar. Now the drawbacks faced in Medlar also is, there are few drawbacks. One is Medlar needs internet connection. If you don't have a live internet connection, you cannot log into your ID, you cannot make a report. One. Far-flung areas, no internet network. So as of now, what is happening is Gangtok network, we have good network, internet network is there. All the majority of the since we just started with Medlar reporting right now, I think in 3 months. So report is coming out in Medlar, number one. Districts, they are still in the, they are not even in the typing phase, they are still in the writing phase. Now from writing phase, at least over here what happened 5 years, 6 years ago when I came, we were already in the typing phase. The reports all comes out typed. Then we pushed on to Medlar online reporting. But in districts, they are still in the, and why, the reason for that is lack of infrastructure is what they say. Yeah, no printer, no computer, no net, type, that was the result. So everything is, they still, till now, north, I think north, west, everyone report is coming in written form. So West Bengal is against Medlar. The state government doesn't want to follow the MHA guidelines, so they will not follow the Medlar or initiatives. So very limited states, Haryana, Rajasthan, I think Sikkim, then maybe 10 to 15 states, I think 10 states we are following that. So the main aim of that is to standardize reporting everywhere. So I forgot your question. What do you think? What did you ask?
Collection of samples, sir. How do you collect the samples?
So this is now reporting of, so collection of samples also, forms are different. All forms like this, all forms are different for state. But I've seen that when you collect the samples, DNA sample form is totally different than the normal blood or other samples which is taken. Like you take a sample, suppose I take a urine sample for alcohol analysis. I don't need to send that to the central forensic lab. Alcohol analysis is done in the, we have it over here only. So problem where it arises is, whenever we take an sample, the two witnesses always will sign. Now consent, it said that sample collection, whether to take the consent also, normally consent is taken, but so many people don't want to give consent. Well, the police will just bring them and they say, isko blood than, so doctor, you are suspected, alleged accused, may not be, he may be an innocent person, alleged accused, so iska blood than. That the problem is he disagrees. Now when a person disagrees, sample collection is difficult. You cannot force. So there the major problem comes. We face that. Over here, what happens is, I think 2 or 3 times this has happened, but it got sorted out. I don't know, after he talked with the police and after 15 minutes he agreed to give consent. Because he didn't stick to that, I don't want to give for a long time, so I have not reached that stage yet. So after 15-20 minutes he gives the sample, he collects, he says. So we just take the sample and whatever preservative is there, like for alcohol we use a grey color vial. If you put it in a red color vial, the sample is gone. It's called a sodium fluoride vial. For alcohol analysis we use that. Any other toxicological testings, we send it in a plain vial or EDTA vial. So there are different vials, colors for each sample, what you want to test. It's not a common vial or something that. So each vial has separate toxicology. I send a separate red vial. Then there's a purple vial which I send for any other tests. Then there's a grey vial. So those samples we give, and the samples is sealed in front of us. The police, either we do help them with sealing. They seal it, and then it goes. The report comes later to the police. Then they forward, maybe they forwarded or sent it to them. But the only thing is, I noticed is 2 witness signature and proper preservation method, that is it. Apart from that, I don't think so there was anything else.
But under CRPC,
Yes.
53A, the medical practitioner can, without keeping any witness, get especially of rape or murder, I'm not sure, rape or any of these sexual assault case, you can get the blood sample drawn, is it?
From the alleged accused?
Yes, yes.
Okay.
The 53A gives you the power, medical practitioner.
Without consent?
Yes, yes.
Okay.
That is
This I did not know, because whenever the form comes, the form itself states there's a box for a witness, it's written witness. So we usually get the witness, we just sign it. So next time if any alleged accused comes regarding this case, we don't need to ask for a
because
consent.
usually what they do is that they come to the court with
Yes.
this blood sample, extraction of blood sample. So what I usually do is that you do not have to come to the court, you can go directly, you can please refer to the section 53A of CRPC and go to the medical practitioner. But they are like, no, the doctors don't want it, they want the order from the court and all that Jabela they do. So I just give them one
Because
simple
thing
order
is,
to comply with that section.
We are not that much expertised in law, so we don't, because everything in OVA is, whenever in medical field, whatever is there, be it any operation, be it any examination, everyone wants consent. Without consent, no one wants to touch, because down the line, there is no harm in taking the consent. They say, China, छोड्दा भयो, बादमा court may say, why did you take without consent? That time, what is the reason I give? That is the thinking of everyone. So whatever process comes, we don't segregate like required or non-required. JMOM consent. So that's what's happening. Anything? Yeah.
For delay is being made on conducting the DNA test, whether it affects the results or not.
Obviously, yes.
But it is done after many years.
It will affect. See, the DNA is all biological material, right? So the longer the time, the degradation has to be, degradation will be there. Contamination will be there. If you don't keep the sample properly, let's assume just a sample is kept in the malkhana, DNA sample, you don't send it for a long time, fungus lagcha, K lagcha, degrade huncha. So faster the better. If you keep it a long period of time, not only DNA sample, you send it like we send for toxicology viscera, you send blood sample for alcohol, anything. Everything, delay is going to affect the results. Alcohol is volatile. The moment you send the sample, maybe the person, you take the sample, fresh sample right now from the urine or blood of the sample, it's a volatile. Test if you do after 1 week, the result is going to show nil, because the alcohol, the evaporate, it will go away. Same thing for toxins, right? It breaks down. So, delay,
So
the
is there
result.
any SOP to be followed with regard to presentation of the sample?
Yes, we have a SOP. In fact, the Sikkim state SOP was made last year. Before that, we did not have SOP. We have, but the thing is, the SOP was made by 2 of us, Dr. Oti sir and myself. We two made it. We made the SOP. We had submitted, because this question was put forth to the Chief Secretary from the Home Department, from the Home Ministry. So till 2000, last year we didn't have a SOP, so we made only the police have their manual or SOP. That's a thick book sort of thing. So forensic, we made a SOP, sample collection, everything. Now the thing is, but the SOP, what we made a file and put forth, but the thing is, the SOP has to get clearance from various departments. It will come to the law, it has to get clearance from the home, then it has to get clearance from the law department, home department, police, court. I think there's we made the SOP and there's a checklist, and the file was put forth last year only. We have not followed it up, but the file may be in process. From our health side, it's clear. No, I have the soft copy. If you want, I can share it, but that SOP is, unless all the departments sign it and agree to it, it will not come in use. It's not used. But I have the copy. If you want, I can share the copy. It's okay.
So in case of drunken driving, so the test has to be conducted within a week after collection.
Within a
Week of collection.
latest. But preferably if better within 24 to 48 hours if it's done, it's better because drunkenness, what happens is main drawback for drunkenness is you are determining the blood alcohol concentration percentage. So if I have 50 or something like that, that doesn't mean that I'm drunk. So there's a chart, right? Above this you are drunk, above this you are so just presence of alcohol doesn't prove that person is drunk. So maybe down the line you take I may be drunk right now, but if you take the sample for testing maybe 7 days later, that percentage may slowly is coming down. So by the time the result is done, the percentage will come in such a negligible value that you cannot prove that person is drunk. That's the drawback for drunkenness.
So what about this DNA collection of blood? Within how many days or how many months the test has to be done to get a appropriate result?
So usually any sample it says that within 24 to 48 hours test got no. That any sample, not only DNA, it's set for all samples. 24 to 48 hours now, when you take out the test, you do the testing for that, any sample. But I don't think so till now, 48 hours ma bhitrama kosko bhako chaina test. So you are sending it to Assam, you are sending it to Hyderabad, you are collecting, you are keeping 2 days in Malkhana, not possible. Even in SFSL also, I am collecting the sample on Friday, so let's assume I am collecting my sample on Friday, and tomorrow is the second Saturday, Sunday, government holiday, SFSL is closed. The person who's the IO in charge, he cannot go and literally open and do the testing also. Usko sample will put on Monday, maximum maybe Saturday, even if he reaches it today, testing is happening after 48 hours. So these are like roadblocks which I don't know how they'll clear this, but these problems are faced, especially during puja vacations. Now the Losung is coming up, SFSL remains closed. So 7 days, 10 days, whatever sample you pick up, testing will be done only after. So that I don't know how they sort it out, but that's the process which is going on. And suppose there is a female victim of rape, right? And usually it's always said that a female doctor should examine, and if a male examines, a female knows that someone is there. Now there is a form says consent of the victim, right? So त्यति बेला पनि
victim we have to take. This is
only for the accused, चाहिँ पढेन. Thank you. So done. Anything else? Last.